Hi, myself Ankit, and hope all of you are doing well. So we were discussing biotechnology, and today's topic is fluorescent in situ hybridization. So this process, fluorescent in situ hybridization, what is this process? See, basically this process helps us to find the location, of a gene present in chromosome. So what the fish, or I may call it as fish fluorescent in situ hybridization. So the fish process helps us to locate a gene present in the chromosome. See, this is a structure of chromosome, and suppose this is a gene I want to find. So I don't know where the gene is. Okay, so by this process we can find in real time that the gene is located at this point or this point or this point or this point, whatever the place it might be. Okay, so to understand the topic we have to understand some background concepts also. So these concepts are complementary nature of DNA. So you know that A is complementary to T and G is complementary to C. And if I take an example of a DNA strand, you know that DNA strand is double-stranded. So there is two strands present, and there are certain number of nucleotides. Suppose this is A, T, G, C, and this will be G, C, A, T. So you might have understood that what is complementary means this strand will be complementary to this strand or the vice versa. Okay, so you now have understand what is the complementary nature of the DNA. Then next concept is the probe. What is probe? Probe is a short stretch of DNA sequences. Probe is a short stretch of DNA sequences attached to any of a reporter molecule. Okay, so if this is a base and this is the sequence, so the probe can be developed in two ways. One, you can directly attach a reporter molecule along with the DNA segment or the nucleotide segment, or you can label these individual nitrogen bases with a radioactive element or any fluorochrome So you can use these elements to modify these bases, and you can develop a short stretch of DNA that is called as probe. So now I understand the complementary nature and the probe. What is the probe and what is the complementary nature of DNA? Now suppose you have to identify a gene that is present inside the DNA. Suppose this blue gene you have to identify the gene where this gene is situated inside the chromosome. So you don't know where these are situated. So what you do, you simply sequence the gene. Okay, you simply sequence the gene using any sequencing method. I have discussed in another video the sequencing methods, that is Sanger's method. You can use Sanger's method, or just for this video, let us assume that we know the sequence of the DNA, and you have already the complementary nature of the DNA. So you developed the complementary strand of this strand. Okay, so if you have A, T, G, C, T, you develop T, A, C, G, A. So you develop this part. By using either radioactively labeled isotopes, or you may have developed them by using a reporter molecule. Okay, so you have developed a probe. Probe is what? Probe is a short stretch of nucleotide sequence that has modified bases. Okay, that the modification can be of radioactively labeled, or the modification can be of reporter molecule. See, reporter molecule is a molecule that reports that something special is in it. Okay, so we will be discussing this in the later part. So you have now developed a probe that is complementary to your known DNA sequence, and you know that DNA is double-stranded, and so another short stretch of strand cannot bind to it unless and until you have converted that double-stranded DNA into single-stranded DNA. So how can you modify this? You can easily modify this by denaturing. By denaturing, you apply enough heat, and the hydrogen bonds between the bases will disrupt and the strands will be separated. So by separating the strand, what you do, you give the single strand DNA a modified isotope, a modified probe. A modified probe has been given to the DNA molecule, and the probe has, as the probe has a complementary nature, it will bind to that specific site only. Okay, and as the probe is radioactively labeled, or a reporter molecule is present inside it, it will somehow let you know that the binding is complete, and you can easily observe the results after binding. If it is radioactively labeled, or anyhow it is labeled, it will emit some lights, or if it has a reporter molecule, then the reporter molecule will release and show some lights, and by under a light microscope or electron microscope or fluorescent microscope, you can easily monitor the changes. Okay, so now you can easily say that okay, so I am getting light from this region of the chromosome, so my gene is surely situated in that part only. So this is how you can locate a gene's position inside a chromosome. So how the process, so I have told you the principle of the process and how the process is done. So you have to extract a cell sample in a test tube. You have put your cell sample inside the test tube. You rupture the cell, and some solutions are added in which the DNA is freely roaming. You now denature the solution. Denaturing leads to the separation of the DNA strands, and now you apply the probes. Now you apply your probe, and then you wait some time and take the specimen in a slide and go into a microscope to observe the results. So this is how the fluorescent in situ hybridization is performed, and the steps follows: generation of probe. We have developed our probe, and then separation, sample preparation. We have prepared our sample. Then what we do the denaturation of the DNA, that is separating the double strands. Then the hybridization occurs. Hybridization means you are hybridizing a DNA strand with a modified base. In this case, it is our probe. And then the results appears by fluorescence or any of the samples that you have taken.